Now let's look closer at the steps of glycolytic pathway. We'll talk about the intermediates of the glycolytic pathway, all 10 chemical reactions for the interconversion, what enzymes catalyze each of these chemical reactions, and the respective mechanism of catalysis. Our goal is to understand the chemical logics behind each step, why each intermediate is formed and what is their meaning, and to understand how overall directionality of the glycolytic pathway reactions is achieved. In the first step of glycolysis, glucose is phosphorylated at 6th position, forming glucose 6-phosphate. The cell plasma membrane is permeable for glucose because of the presence of glucose transporters. This way, glucose can go freely in and out of the cell. However, there are no transporters that can permeate glucose 6-phosphate. This way, important function of the first step is to trap glycolytic intermediates in cell cytoplasm, thus making it available for next steps of glycolysis. The transfer of phosphoryl group to glucose, making the phosphoester, is an endergonic reaction because this ester bond between glucose and phosphate group has some free energy. So to make this reaction energy favorable, the phosphate group is donated from ATP, where it is even more energetic than the phosphate group transferred to glucose. This way, whole energy balance in the reaction, energy drop between the substrate and the product is highly negative. At standard conditions, the energy drop, energy release of the first step of glycolysis is very high. This way, the first step of glycolysis is virtually irreversible chemical reaction. The phosphorylation of glucose is catalyzed by an enzyme called hexokinase. Hexokinase uses magnesium ions as cofactors. And as we will see later, all the enzymes which operate on phosphointermediates usually have magnesium as a cofactor, which helps to coordinate highly negatively charged phosphogroups. Mechanism of hexokinase action is the induced fit. For details of hexokinase catalysis, I would refer you to the Langer chapter describing enzymes, where hexokinase catalytic mechanism is described in detail. Altogether, in the first step of glycolysis, glycolytic pathway starts with 1 glucose uses 1 ATP converted into ADP to produce 1 glucose 6-phosphate. In the second step, the glucose 6-phosphate is converted to fructose 6-phosphate. And aldose is converted to ketose by an isomerization reaction. The purpose of the second step is to make glycolytic intermediate a more symmetrical molecule, which makes it better prepared for cleavage into almost identical pieces. The reaction is almost neutral from the energy perspective. The free energy difference between substrates and products is only around 1 kilojoule per mole. So this reaction is entirely reversible. The isomerization reaction is catalyzed by an enzyme called phosphohexose isomerase, which uses magnesium ions as cofactors. Phosphoglycolysis isomerase can perform a similar aldose to ketose isomerization reaction also in the case of other hexoses, not only glucose 6-phosphate. The mechanism of phosphoglycolysis isomerase catalysis is an acid-base catalysis. The job of enzyme is to move two hydrogen atoms from the second position to the first position, thus converting first carbon from aldehyde to alcohol and second carbon from alcohol to carbonyl group, keta group. The critical function of the enzyme relies on glutamate residue, which performs nucleophilic attack on the hydrogen atom in the second position and converts the substrate to highly unstable cis-enediol intermediate. Cis-enediol intermediate then collapses to a final product, fructose 6-phosphate. In the third step, the fructose 6-phosphate is further phosphorylated in the first position to become fructose 1,6-bisphosphate. As in the case of glucose phosphorylation, to gain energy required for formation ester bond between phosphogroup and fructose, the phosphogroup is transferred from highly energetic ATP converted into ADP. This way, the overall energetic of reaction is highly exergonic, so the in standard conditions approximately 15 kilojoules per mole of energy is released, free energy is released. This way, the reaction is a virtually irreversible chemical reaction. It is catalyzed by an enzyme called phosphofructokinase 1 or PFK1. PFK1 uses magnesium ions as cofactors and relies on a very similar mechanism of induced fit similar to the hexokinase. The step 3 is also called the commitment step. At this step, cell decides whether it wants to burn more glucose in a glycolytic pathway or not, because this step is irreversible. Because of that, phosphofructokinase is a highly regulated enzyme. It is regulated by energy availability through the allosteric regulation by ATP and other metabolites sensing cellular energy balance. In step 4, the 6-carbon backbone of fructose 1,6-bisphosphate is finally broken down into almost identical 3-carbon pieces, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is highly energetically unfavorable at standard condition. The standard free energy change in this reaction is approximately more than 20 kilojoules per mole. This reaction does not occur at standard conditions. However, it is fully reversible at cellular conditions. The reason is that actual free energy change in the chemical reactions, it is a combination of standard free energy change at standard conditions and a contribution of mass action term, which is determined by actual concentration of substrate and product in the chemical reaction. In step 4, the previous steps, step 1 and 3, are highly exergonic steps. They pump a lot of substrate into the left side of the reaction. The following steps after step 4 are also highly exergonic steps. They constantly evacuate the products of the step 4 reaction. This creates a high concentration difference between the substrate and the product. This in turn drives the mass action term highly negative, which compensates the highly positive standard free energy change. This way, the reaction in actual cellular conditions becomes fully reversible. The reaction is catalyzed by enzyme called aldolase, and this is a reverse of aldol condensation. The catalytic mechanism of aldolase is quite complex, and it is an example of covalent catalysis. In a critical step, the lysine residue within enzyme active sites form a Schiff base with the second carbon in the substrate. This destabilizes the carbon backbone sufficiently to break it apart into 2 3-carbon pieces. As such, the input in the step 4 6-membered 6 residue 6 carbon atom glucose 1,6 bisphosphate is converted to the 2 3-carbon pieces dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Step 5 completes preparatory phase. The output of step 5, which is utilized in payoff phase, is glycerol 3-phosphate. The purpose of step 5 is to convert remaining half of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, making it suitable for the follow-up steps. This reaction is endergonic at standard conditions, so it is energetically unfavorable in standard conditions to convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Similar to step 4, this reaction is made fully reversible due to mass action. The product of the reaction, glyceraldehyde 3-phosphate, is constantly evacuated through subsequent steps, thus driving the mass action term negative that compensates for a highly positive standard free energy change at standard conditions. A result of this reaction is the production of 2 identical glyceraldehyde 3-phosphate pieces, one of which originates from the first 3 atoms of fructose 1,6 bisphosphate and the last from the last 4 atoms. The reaction is catalyzed by 3-phosphate isomerase, and the mechanism of the catalytic mechanism of this enzyme is similar to the mechanism of hexose-phosphate isomerase that we discussed at step 2. So this concludes the preparatory phase of glycolysis.